Hi, good afternoon. My name is Mehmet Toner. I trained as an engineer at MIT some years ago and then moved to Mass General Hospital, Harvard Medical School across the river in the Department of Surgery, and I run a microfluidics and microelectromechanical systems laboratory, and it's a national resource center funded by NIH, and our goal is to find meaningful clinical applications of microtechnologies and also translate them, having access to a clinical environment. So within that, we have the microfluidics is extremely powerful technology. It gives you an unprecedented ability to manipulate cells on chips and devices at a single cell level and in a very high throughput fashion. And you've heard some very exciting examples from my colleague, Dr. Jehan, a few minutes ago on how to isolate rare cells using microfluidic type approaches. And we have focused on a number of exciting technologies and learned in the process. And we did this from a clinical perspective. The way we look at the problem is there's the blood test, which is the most common test, ubiquitous test used in medicine. But it's not very sensitive. For each blood test, a set of values, you could have 100 different diseases that fit that. And then we take people, put them into big boxes and radiate them and get much more sensitive, specific information, but then it's expensive and invasive. On the other hand, the blood contains far more information than we have used. You can almost virtually diagnose and monitor every single disease, from bacterial infections to cancer to Alzheimer's, using various particles in blood, whether they might be specific rare cells or viral particles, bacterial or exosomes. So we are using these microfluidic technologies to manipulate these particles in ways that we can provide useful clinical information. And a number of technologies we have developed are one we call a pachinko device, and it is like the Japanese game pachinko. They could separate blood into its individual components based on their sizes. It's an extremely, the blood will come from one side, and there are a number of barriers in front of the blood, microscopic micro-etched barriers. And as the whole blood goes through that, smaller particles will easily navigate through these barriers, whereas the slightly larger particles will be deflected because of the differences in the radius. And this way you can almost like we can create this very sensitive size separation where you could get red blood cells, platelets, and buffy coat or neutrophils and monocytes and different size cells, very high throughput fashion, and that has a number of different applications I'd like to mention later. But the idea of size-based separation using these high throughput microfluidic devices is a very exciting area. Another area that was significantly covered by, again, my colleague Jehan is what we call inertial focusing. And this was originally discovered in our lab by Dino Di Carlo, who's now a faculty member at UCLA. And what happens is when you are in a laminar flow, it's a fancy name for a flow that is no mixing, and particles sit in a streamline, and they really don't move between streamlines. And what we have found out that as you lower the channel size, so the particle size is at the same orders of magnitude as the channel size, then different forces start acting on the particle. One force is that shear gradient pushes the particle towards the wall of the channel, and the wall says, no, don't get close to me, that has energetic cost to you, so it pushes back, and the particles suddenly shift from their streamline and find extremely predictable locations in these channels. Basically, they self-sort. And this self-sorting mechanism is extremely important. And if you also put, as Jehan mentioned, Dean forces, which is basically curvature, you increase the number of forces on the particles, and you can really have billions of blood cells go through these channels at speeds that we have achieved now up to 10 meters per second. You're talking crazy speeds, but these particles moving, so it's not damaging to particles or the cells. And you can sort them based on their size, you can sort them based on the viscoelastic properties, and you can align them so that you can interrogate them, optical means or other chemical means. So it gives you, again, a very precise manner of orienting the particles and interrogating them, as well as using their intrinsic properties to separate them. So that's another area where we are exploring a lot of different applications. Again, I will mention some of those later. And another technology we developed is what we call immunoaffinity isolation of specific particles. So here, now you're getting a much more specific. If a particle, a cell that you're interested in has surface properties, an antigen, you could put a glue on the surface, an antibody that will bind to that cell. And if you control the microfluidic environment of this, you could actually, in a whole blood, for example, in the case of circulating tumor cells, we have done that, you could have every particle, and remember, in a tube of blood, you have about 100 billion cells. And if you're putting that through a microfluidic device within a few hours time period, 100 billion cells are going to interact with the surface if you make the mixing in that device very predictable. And if there's a rare cell or cell of interest, a target cell, an infected cell, and you have an antibody for it, it will bind to that surface. This way you can isolate particles much more sensitively than max or flow cytometry type systems that are not developed for these kind of clinical applications. And so these are some of the platform technologies we have developed that has significant different applications. And the beauty of it is that we could process tiny little amounts of blood, a finger prick of blood very precisely, as well as a tube or 2 tubes of blood. Our current speeds now, we are up to several tubes of blood within an hour. So we're talking about hundreds of billions of cells processed and interrogated within an hour. And one of the earlier applications we explored was counting Cd4 cells, point of care, very precisely within minutes. This is very important for global health. If you could take a fingerprint of blood, put on a chip, and it tells you how many CD4 positive T cells, because that's how you determine in global health applications whether or not you should treat somebody with antiretroviral drugs if they have AIDS. And this monitoring was for World Health Organization was one of the #1 technical issues. And flow cytometry or optical ways of doing that is not feasible in resource-limited areas. So the microfluidic chip we developed in the academic lab, now it's a spin-off company called Diagnostics, takes a fingerprint of blood, it meters exactly 10 microliter, puts through a surface that captures with CD4 antibodies, CD positive for T cells. But because the fluid flow is so precisely controlled, monocytes that have CD4 also on their surface, because of their number, expression level and the size, they don't stick to the surface. You can very differentially capture T cells and not monocyte. So it's a label-free, very quick testing of CD4 T cells in blood from a small finger prick of blood. So there you see a very powerful application of these kind of tools. Another one that we are, since the mid-2000, first paper we published in 2007 is circulating tumor cells. We know cancer spreads from the peripheral blood, but these are extremely rare cells, maybe 1 in a billion blood cells. You have a cancer cell. How do you interrogate enough blood cells, blood volume, to find enough of these rare cells that then you can clinically use to monitor? We can go and sequence these cells and PCR them, so we can look into specific mutations. And today the cancer treatment is going almost exclusively towards targeted therapy. So if you have certain mutations in lung cancer or melanoma, you're put on specific treatments based on that mutation. And this will be a liquid biopsy, non-invasive way of getting the information from blood and clinically actionable. So you could monitor these patients, you could look at their genetic mutations, makeup, and give the right drugs to the right patient at the right time. We have also shown that we could detect these cells in circulation very early. In the case of prostate cancer, for example, in localized prostate cancer, we can find these cells. And that has tremendous potential, of course, and in the long run, can we do this as part of an annual checkup as a screening. And so these are the type of applications this kind of technology could have in medicine, in circulating tumor cells, and we are exploring many of these. The more futuristic ideas, because the power and we more understand how to push cells and blood and bodily fluids on these chips, and I call it putting a garden hose through a microchip, because you're processing very large volumes of blood. People have not been able to do that until very recently. It really opens up in all kinds of different possibilities beyond the ones that we discussed today, such as processing peritoneal fluids, ascites, or other types of urine to find, again, very precise the cells of interest and bacteria that could have implementation. For example, we are using inertial focusing, as also Dr. Jehan mentioned. Can we isolate fungus from blood so that we can, because fungal infections are tremendously important in clinical medicine and surgical wards, and can we very quickly identify fungal infections from large volumes of blood without the need of complicated culture techniques and whatnot. So we feel that as we explore and learn this territory, we will have far more precise and clinically actionable diagnostic tests that will help clinicians to monitor and manage their patients better. So that's our vision and our hope, and thank you very much for the opportunity.